Assalamualaikum and hi everyone. My name is Salwana Arina binti AKHIR. My metric number 23DBT 20F2005. Today I'm going to show you guys about purifying protein by chromatography. For your information, protein purification is a series of processes intended to isolate one or a few proteins from a complete mixture, usually cells, tissue, or vast organism. Protein purification is vital for the specifications of the function, structure, and interaction of the protein of interest. Next, about chromatography is based on the principle where molecule in mixture apply onto the surface or into the solid and fluid stationary phase. Stable phase is separating from each other while moving with the air of a mobile phase. Firstly, place the E. coli tube in the microcentrifuge for 10 minutes to separate cells from the growth medium. Remember, the tips have to place composite to prevent damaging the message. After 10 minutes, remove E. coli tube from microcentrifuge and dump out the supernatant carefully. Second, we have 2 pellet additional cells. Then set the P 1000 to 1000 microliter and attach a tape. Next, withdraw out 1000 microliters from the EC2 tube. This is to add more cells to the EC1 and also to increase the amount of red fluorescent protein. After that, inject the tip into the biohazard waste container. Next, we have to intervene EC1 tube for 10 minutes. After 10 minutes, the EC tube should be removed. Third, choose P200 by micropipette and set it to 150 microliter with a new from this box. Using the micropipette, transfer 150 microliter of elution buffer to EC1 tube and as always, eject the taps to biohazard waste container. Next, vacuum the EC1 tube for 10 seconds to resuspend the cell patterns. Attach a tip to the P200 micropipette to transfer 150 microliter of lysis buffer into the EC1 tubes. To prevent the contaminations, eject the tips into biohazard waste container. Then continue with centrifugate the easy one tube as the step we did in step 1. Balance it and wait for another 10 minutes. Then set the timer for 12 hours to incubate EC1 till overnight at room temperature. Continue the purifications by placing the waste collection jar under the valve and drains away the liquid in chromatography column. Centrifugate the EC1 tube for 10 minutes. Then set the P200 micropipette to 200 microliter with a tip by using the micropipette. Transfer 200 microliter of supernatant from EZ1 tube into super tube. Then attach a new tip of binding buffer. The binding buffer allows the highest DNA binding of any column method. The binding buffer is then dispensed into the super tube. The tips then ejected into the biohazard waste container. Next, apply the soluble fractions to the column. Then, we pick up the P1000 micropipette and attach a P1000 tips. Next, we draw out 400 of the super solution. The red super solution is dispensed into the chromatography column. Then, ejected into the biohazard waste container. Place the waste collection jar back under the valve. The column is open and allow the liquid to drain into the waste container. The valve is then closed when the liquid is just above the resin bed. This will release hydrophilic proteins. Then we wash the column. Set the P1000 micropipette to 1000 microliter and attach a new tip. Dispense 1000 microliter of the wash buffer solution into the chromatography column. This is used to separate out unwanted proteins. The tips is ejected into the biohazard waste container. Next, close the valve is then closed when the liquid is just above the resin bed. This will release moderately hydrophobic proteins. The column is open and allows the liquid to breathe into the waste container. Lastly, include and collect the RFP protein using a new tips. Then draw up 1000 microliter of the elutions buffer solution. Dispense gently down the side of the column and repeat this step. This will release red fluorescent proteins. The column is opened by turning the valve. Wash the red protein revealing through the column. When it's near the bottom, the valve is closed. Then, the open RFP tube is placed under the valve. The column is then opened to capture the RFP. The valve and RFP tube is closed. The waste collection jar is placed back under the valve. 1000 microliter of the column equilibrations buffer CEB solution is dispensed into the chromatography column. Equilibrations buffer provides conditions to ensure that the target molecules interact effectively with the ligand and are retained by the affinity median as all other molecules wash through the column. Then we open the column and allow the liquid to drain into the waste container. Close the valve when the liquid is just above the resin bed. Repeat 3 times before storing the column away. Okay, you guys can see this is the final result for this simulation.